thank you everybody for joining tonight. I have to say that one of the silver linings behind all of the restrictions over the past year and a half is that it has really forced us to engage with the available technology for doing online meetings and presentations. And what that does mean is we can go a little bit further afield than we normally do. Being in the far north of Scotland, it does pose problems for guest speakers travelling the length of the country. But by the wonders of technology, I'm delighted to say that we've got Dr Martin Braddock joining us this evening during our session to give a talk entitled Extremophiles and Where There Must Be Life Elsewhere in the Universe. So I'm just about to hand over to Martin to begin this talk in a moment, but just before doing that, I'll provide a few basic details. First of all, tonight's talk is being seen on Zoom by members of TESS Astronomy Group and also by members from North of Scotland astronomical societies. So we've got the Highland Astronomical Society, we've got Sigma, Aberdeen Astronomical Society, Cairngorms Astronomical Society, and I'm not seeing particular names there, but there may well be North Ronaldsay Astronomical Society, so that's the most northerly out of the bunch. So obviously it would be quite difficult for Martin to get round all of these different groups, even though we consider ourselves to be relatively close, all being in the north of Scotland. There is still quite a travelling distance between all of the groups, and even worse, if Martin was to travel here, I dare say it would take you a whole day just to get to this end of the country, and then a whole day travelling back. So it's good that the wonders of modern technology are allowing you to come and join us this evening, and we can enjoy your presentation from the comfort of our houses without having to travel at all. So I think that's probably a good point to hand over to yourself, Martin. Martin is going to do a bit of introduction of himself, simply because he does an awful lot in terms of STEM and other things, and it's probably better if he picks the most pertinent things from his very impressive CV. So I'll hand over to Martin now, and after Martin's talk, there will be time for questions and also a little bit more TESS Astronomy Group news briefly at the end. So over to you, Martin.
Thank you very much, Gordon, and thank you also, Christian, for setting this up for us. And also apologies, as it was my suggestion to reschedule it, but when I saw that England and Scotland were playing on the same night, I thought, you never know, that might just be a distraction for people. And even though it perhaps wasn't the most exciting game, I'm sure it drew a large audience. So thank you very much for accommodating that too. So just by way of an introduction to me, professionally, I work in drug discovery and in drug development, and have done for about 25 years. And prior to that, had an academic career at University of Oxford, working at that time in a very topical area, the HIV viral area. And since then have moved on and worked in industry in various phases. But why I'm particularly interested in space and anything astronomical is there is a real need for us to understand the possibilities of discovering and developing new medicines that could be applicable to the space environment. And one of my real interests, one of my many real interests, is in understanding the effects of microgravity on the human body and some of the types of studies that can be done on the International Space Station, and were actually done in the predecessor to that on the space shuttle, and also on some of the Russian space missions. So just by way of my background, I'm a scientist by training. I worked in biochemistry and in radiation biology. I also now have professional qualifications in astronomy, which is a joy of having a hobby as well as a job. And I really do enjoy, I hope, giving a different facet on the science that we all love as amateurs or maybe semi-professionals. And what I want to inspire you, I hope, with tonight is the topic that I've got up here, extremophiles, and why there must be life elsewhere in the universe. So the introduction here actually shows a particular extremophile in the top left, which is a bacterium that we'll come on to a little bit later. But in the bottom left, and we'll come on to this in more detail too, is a tardigrade, which is probably one of the ultimate extremophiles. And on the right-hand side is a linear accelerator. So just keep that in your mind for now, and I'll explain why that's on the front cover of this presentation. So what I'm going to go through is a little bit about life in the universe and why do I believe, and I hope most people believe, why is life likely elsewhere? Introduce extremophiles and some quite astonishing examples of organisms that can withstand extreme environments, one of which was only published the month before last. Describe a little bit around biosignatures as signs of life, and then summarize our findings to date, and of course acknowledge people who have helped me over the years and challenged and had input into thinking, including the sharing of pre-published work, for which I'm very grateful. So how I've tried to pitch this is being scientific, but not too scientific, so suitable for people who don't have a strong scientific background. If you do have a strong scientific background, there's something in there for you too. So I hope I've got the right level. So just by way of a start, and I'm sure you've seen this before, this is something that really piqued my interest when I first came across it, probably about 15 years ago. It's one of Arthur C. Clarke's wonderful sayings, which is two possibilities exist. Either we are alone in the universe or we are not, and both are equally terrifying. And perhaps when he first made this statement, we didn't have perhaps the realization that we have today that, for example, climate change is real. And if we are alone in the universe, as Carl Sagan said in his immortal The Pale Blue Dot, there is no one to come and save us. And actually, it's up to us to make the best of what we've got. If we're not alone in the universe, then of course it begs the question, as we all perhaps theorize, fantasize, hypothesize over what type of life might there be in the universe, and what type of life might we be able to find in the solar system. So some very early thinking, and this tickled me a little bit. So this is Metrodorus, round about 330 before Christian era, and of course this is translated from the Greek at the time. But it's an interesting statement that even in the 3rd century BC, there was the thinking that to consider Earth as the only populated world in infinite space is so absurd as to assert that an entire field sowed with millet, only one grain will grow. And of course, that's a pretty brave thing to say in the 3rd century BC, and probably even more of a brave thing to say in the 17th century and 18th century, when this French philosopher stated that intelligent beings exist in other worlds, for instance, the moon. If this were the case, the moon's inhabitants would already have come to us before now. So these are just two examples of the many that there are in the records, where philosophers at the time were already thinking about life beyond Earth. And of course, this is almost at the bottom here with Bernard Lebouvier de Fontenelle, almost, if you like, the Fermi paradox, which we'll come on to in a little bit. So that got us thinking, I think, about some scientists that we need to know. And again, you may be familiar with some of these. You may not be familiar with any of them, but that doesn't matter. I'll introduce them as we go. Stanley Miller and Harold Urey, who conducted a landmark study that I'll describe in a moment. Charles Cekell, based in Scotland, University of Edinburgh, one of the leading lights in astrobiology, or the field of astrobiology, and Sarah Seager, based at MIT, Massachusetts Institute of Technology in the United States, also a very leading light. And as two examples of really scientists right at the top of their field in the discovery of exoplanets, but also in thinking about the possibilities of life. And Charles publishes much of his work on open access papers. So unlike some people where unfortunately you have to pay to access their scientific work, Charles publishes most of his on free open access publications. So if you're interested, please do a literature search on Charles Cekell. And if you've been lucky enough to have him talk to your society, then hopefully you'll confirm what I've just said. So this was the experiment done in 1953, the Urey-Miller experiment, or it's sometimes called the Miller-Urey experiment, depending on which textbook you read. And this was revolutionary at the time, so much so, of course, that in the 1950s, there was still a very, very dominant religious philosophy around the world, particularly in the United Kingdom, where anything that could go against the idea of life generated by a creator was really seen as challenging the establishment. And to some extent it succeeded, and it succeeded because very simply it showed that under conditions that tried to mimic the early Earth, give the very simple molecules that were hypothesized to be around at the time, water, ammonium, hydrogen, methane and carbon dioxide, and lightning, or lightning simulated by electrical sparks, and the right type of atmospheric pressure, or at least predicted atmospheric pressure, it was possible to produce some very primitive amino acids and nucleic acids. And that was almost seen as justification that life could be formed. And of course, the reality is we're not producing life here, we're producing some of the very basic building blocks of life. But nevertheless, this was a revolutionary experiment in the 1950s. And what was subsequently conducted a little bit later and then published in 2011 is that it was really quite flawed to some extent, because the early experiment didn't quite accommodate what was subsequently believed to be the reducing environment on Earth. So it was repeated. And when it was repeated, not only did it verify the findings of the Urey-Miller experiment in the 1950s, but it actually showed that there was an even greater complement of amino acids formed. So in a way, it substantiated the original study, but it also showed that a greater repertoire of amino acids could be formed. So very interesting study. And of course, oops, oh, I got stuck. Oh, there we go. So relative abundance of the elements, and why is hydrogen, carbon, nitrogen, and oxygen-based life likely? And I think we all know this, that to a certain extent, it's a thermodynamic property of the way that atoms form and molecules form. And we get to a certain point where we need to create so much energy in supernova explosions to produce the heavier elements that we're almost, to a certain extent, biasing the population of molecules in the universe to be these simpler, low molecular weight molecules. So of course that includes hydrogen and helium, as we all know, but carbon, oxygen, nitrogen, magnesium, and eventually we can get up to phosphorus, although that is a little bit outside this molecular weight. And of course, what can you make with all of these? Well, we can make simple sugars, we can make lipid membranes, we can make nucleic acids either in single-stranded form or in double-stranded form. And of course, we can start to make some very early proteins. But when you put all this lot together, well, actually, what can you really make? And of course, what you could really make is life. and the concept that perhaps life started as an RNA world rather than a DNA world has a lot of support in the scientific literature. So when we think about that, then it becomes almost, to me, an illogical argument to argue against the formation of life on other worlds. Because if our argument is predicated on the existence of some small molecules in the right concentration at the right time and under the right conditions, it stands to reason that there have to be other planets or other bodies, maybe not planets, other bodies, either within our solar system or within the universe that either have or can support life. So some examples of extremophiles, and I'll go into some more details later. And we may be familiar with some of these, you may not be familiar with some of these. I'll bet there are a couple that I'll show you later on that you won't be familiar with. So we know that extremophiles can tolerate a wide range of temperatures, and they exclude freezing temperatures, and I mean really cold temperatures, not just 0 degrees, but really cold temperatures, right the way up to some intensely high temperatures. We know that organisms will tolerate wide ranges of pHs. So acidophiles and alkalophiles will tolerate low and high pHs respectively. There are also organisms that will tolerate different saline concentrations, so different salt concentrations, and can either be moderately halophilic or extremely halophilic. And then there are also organisms that will tolerate a wide range of pressures, and such that are found at the bottom of the oceans or even high up where the atmospheric pressure is reduced. So you can find, as we know on Earth, pretty much everything. And of course, David Attenborough and other naturalists have done a fantastic job over the decades in cataloguing the wide range of organisms that we can find on Earth. So some of the examples, and again, I'm sure you've seen some of these before, and I've been lucky enough to see bacterial mats in Yellowstone. That wasn't my photograph, unfortunately, but I've seen bacterial mats in Yellowstone. And if any of you have been there, you would not imagine that anything could grow in that environment. Now, black smokers that are produced towards the bottom of the oceans at great depths, again, in an environment that's sulfur rich, that's warm, and that's steam that's coming out of these fumaroles at the bottom of the oceans. Life is abundant around these, around these structures. And then on the right-hand side here, this is just another example of life found in Pitch Lake in Trinidad. So this is a lake that is actually producing pitch oil, and there is life, abundant life in this oil. So life can tolerate a wide variety of extremes. And there are some other examples here that I've included, and there are two that I think well, there are even more, I'll show you in a moment, but two in particular that make me smile a little bit when you look at them. So an algae that can need almost no water and be found at great depths within the earth. And then this fascinating organism in the bottom right, a loriciferan, which is a sea-dwelling creature that lives virtually without oxygen. So it's right at the bottom of the ocean. The partial pressure of oxygen is so low, it's virtually non-existent. And yet this organism can obviously thrive and reproduce quite happily because that's where it's found. So there are a couple more examples, and there's one that I'll go into more detail in a moment. So this beautiful little beetle at the top here, red flat bark beetle found in northern Alaska, has antifreeze proteins in its blood. And for any of us old enough to remember the Liebfraumilch that allegedly had polyethylene glycol in it, antifreeze, that always makes me smile a little bit because I just wonder whether or not any of those beetles had found their way into any of the vines at the time. But I'm sure they didn't. So that's one particular organism that's able to withstand cold by producing antifreeze proteins in its blood. The one in the middle, Deinococcus radiodurans, is a bacterium which can withstand astonishing doses of radiation. And I'll tell you more about that in a moment. And then there are these endoliths that can live in rock up to 3 kilometers below the surface, and apparently able just to live off the minerals in the rock themselves. So the prize, I think the prize goes to this one, which is the tardigrade, which was shown on the opening slide. These can withstand absolutely astonishing temperature ranges, minus 328 degrees Fahrenheit up to 304 degrees Fahrenheit. Fantastic levels of pressure, up to 1200 times atmospheric pressure. They can withstand desiccation, as you can see here, for nearly 10 years, and they can withstand massive doses of radiation. So if you're not familiar with the GY, which stands for a gray, 5 to 10 of them would kill us. These can withstand and thrive in 5,000 doses, 5,000 gray of ionizing radiation. They're found in water, and they're commonly found in water. They're perfectly harmless to humans, but they do eat moss and other bacteria. So actually they're part of the, if you like, they're part of the lower food chain. So I thought I'd mention again these in a little bit more detail. So Deinococcus radiodurans, unlike the tardigrade, if you thought 5,000 gray radiation was high, these do take the biscuit because they can withstand 15,000 gray of radiation. And this is something that's always puzzled me, because that dose of radiation is not found naturally on Earth. So just file that in your minds for a moment. Now, one of the properties of these organisms, of course, is the ability to withstand these radiation doses. But how do they do it? And there was a race on in the 2015-2016 timeframe, where a number of groups were trying to find what was it that made these organisms resistant to radiation, and of course, were there any applications? And the first lab to conduct studies were in the Kunieda lab in Japan, and they found the protein, or one of the proteins as it's now known, but they found what they thought was the protein at the time, that they called DSUP, which stands for damage suppressor. And they cloned the cDNA and they expressed the protein, and they thought, what can we do to show whether or not this protects against the effects of radiation? And on the right-hand panel, these are human cells that have been transfected with this protein, or this cDNA, which expresses the protein, and then human cells have been expressed. And on the bottom, in the sort of turquoise color, as the dose of radiation increases, the number of cells die. Sorry, as the time increases, the number of cells die when they've been irradiated at 4 gray. And at the top, cells that have had the transfected DSUP protein, not only do they continue to grow, but you can see that their cell count actually increases non-linearly over time. So this is a fascinating study, because this is telling us that there is a protein in this bacterium that we can isolate and we can put into human cells that is making these cells, if not radio resistant, at least more tolerant to the effects of ionizing radiation. And why this is so interesting is, first of all, it conjures up all sorts of studies about, could we make human beings radiation resistant in future? And that's to some extent the human enhancement field, and that's not what we'll talk about today. But one of the areas that's of immediate relevance to us on Earth is thinking about, well, is this a property that in some way we can interfere with in cancer patients who receive radiotherapy and whose tumors are resistant to the effects of radiation? Is there something that we could disrupt in this pathway that may make their tumors more sensitive to radiation, or at least take away the resistance to it? And this has real applications for cancer patients who need radiotherapy on Earth. So not only do I see here a fantastic example of the science behind extremophiles, how we can use extremophiles, but also the possibility to do good for patients on Earth. So you might be thinking, well, what does this protein do? Does it sort of put some magical shield around the bacterium? And of course it doesn't. What it actually does is it allows the DNA in this bacterium to be repaired very, very quickly. So most of the effects of ionizing radiation on DNA are to cause breaks in the DNA. And what this protein does, and there are now other proteins that are stimulated, and they repair the DNA very, very quickly, thus allowing the bacterium to live. So it's been a fascinating scientific area, and this has really only happened within the last 6 to 7 years. Oops. Okay, so I wanted to show you something else, because I don't know whether you've seen this before, but again, this stimulated my interest. So this is from 10 years ago now. So hypergravity, I think we all know what hypergravity is, but just in case anybody is less familiar with it, it's really where the force of gravity is greater than that on Earth. So greater than the 1G that we experience on Earth. And what this study investigated was the effect of hypergravity on a number of organisms. And lo and behold, they found that 5 very simple organisms all survived and grew at up to 72,000 times Earth gravity. And to cut a long story short, an organism called Paracoccus d nitrificans actually was able to grow in excess of 403,000 times G. And again, if you remember the seed I tried to plant in your mind about bacteria that could withstand levels of radiation not normally found on Earth, 400,000 times G is not found on Earth. There is no condition today that we know of that would generate that G force. And it is only found in cosmic environments, such on massive stars and in the explosions of massive stars, or in the shock waves of supernovas. So those are 2 examples, perhaps, of where life on Earth actually has evolved, if we believe it's evolved, to tolerate conditions that actually aren't found on Earth. So file that away for a later. And this is just to show you actually the studies here, and these pellets at the bottom just show the fact that bacteria were able to grow and survive. And I think there's another build in here which shows that for P d nitrificans at 403,602 7 times G, it's able to grow. Absolutely astonishing. And I think this again is just more data that supports that. And again, for comparison, there are a number of other organisms, which includes E. coli, Escherichia coli, and this is a Salmonella. This is Saccharomyces cerevisiae, which is yeast. It's not an unusual property to see bacteria and even eukaryotic cells with Saccharomyces cerevisiae that will tolerate these effects of hypergravity. So this is the publication that I think it was in May this year, that again, as soon as I saw this, I thought, wow, this is astonishing. So if you remember back to the tardigrades again, these are our little water bears. These for me have the ultimate prize. Well, there are 2 more prizes that they add. One is that they will actually survive living on the outside of the space shuttle. So they won't, of course, enjoy all the conditions that we find inside, so they'll live outside the space shuttle. Lo and behold, these investigators studied in the linear accelerator that I showed you on the front page, what would happen to a tardigrade if it was accelerated into a sand block. And they found that at nearly 800 meters per second, these tardigrades would survive that impact. 800 meters per second. And of course, the implication is it possible that tardigrades that have come from beyond the Earth may, of course, have evolved to be able to withstand this type of impact. And the publication, again, and I'll leave it here just in case anybody wants to make a note of it, because this is free to access as well, open access. It does suggest perhaps that at least in some instances, there may have been the provision for tardigrades to come to Earth by some form of impact, and if not come to Earth, at least perhaps to have come to other icy worlds. So some very recent publications, and again, if you're interested in them, of course, as this is recorded, you'll be able to access them. Most of these are open access. The one at the top really piques my interest, of course, the extremophilic pharmacy, drug discovery and the limits of life. What can we find from extremophiles that may tell us about extreme environments that may allow the production of certain proteins that could have pharmacological value? And there are a whole a range of publications here, most of which have been in the last year or so. So extremophiles condition in space, I think we're all fairly familiar with this. The low gravity may cause DNA in cells to mutate. It will certainly affect membranes. High levels of radiation, again, will affect the DNA, induce both single and double-stranded breaks. And of course, we benefit by the protection of the ozone layer. Of course, there's no oxygen, so it's a problem for aerobic organisms, but perhaps not for anaerobic. And then, of course, we have the fact that there are extremes of temperature. So this led to one of the studies that Charles Ceckel conducted some time ago now, and he called this the Space Survivor. And this is taken from one of his publications. He went down to Beer on the south coast of England, and he isolated a number of organisms in a rock structure here. And he was able to have these strapped to the outside of the space shuttle. And apologies, I think I said the International Space Station first time round. There have been some studies on the ISS, but this was on the space shuttle. And then what they conducted was a study for just over 540 days, 548 days. And having put these, this is now going back to the study on the space shuttle. They now conducted this study aimed specifically to understand what would happen to this cohort of bacteria subjected to UV radiation, extreme desiccation, and ionizing radiation. And it was between February 2008 and September 2009. And what came back was a cyanobacterium. And the bacterium that came back was quite a specialized part of the phylogeny of cyanobacterium. And one of the things that Charles and his co-workers commented on at the time is this was believed to be one of the earliest forms of bacteria found on Earth. So of course the supposition was, is it possible that the early life forms on Earth either evolved on Earth to accommodate the conditions of the time, or is it possible that these organisms may have survived in space or come either from comets or from other planetary bodies that impacted Earth? So again, it seeds in the mind, is there the possibility that life has tolerated these extreme conditions in space and may at least have instigated some form of primitive life, seeded life on Earth? So I want to come on to a little bit now about thinking about life in the universe, and I'm sure everybody's familiar with this. This is the Fermi paradox. This is a little bit like the Fontenelle question right at the start of the presentation. Why hasn't anybody come to visit us from the moon? And of course, Enrico Fermi asked the question, well, where is everybody? It's called the Great Silence, or Silentium Universae, which sounds much more sinister. And the hypothesis is very simple. Given the age of the Earth, the age of the Sun, given the fact that we can at least model what it might take, and of course this is a big supposition, what it might take for an advanced terrestrial or technical civilization to colonize the galaxy. There's been ample time for it to happen. So basically, why haven't we been visited by anybody before now? And of course, the answer is very simple, or it might be simple, and it's made even more perhaps perplexing when we look at the Drake equation. And again, apologies for everybody who's familiar with this, but if you're not, it was a very simple effort in the 1960s by Frank Drake to try and understand how many potential sources of life there may be in the universe. And if I put everything in, what he's worked through is the number of stars in the Milky Way. And of course, there's a lot of supposition here. Fraction of stars that have planets around them, number of planets per star capable of supporting life, Again, a lot of hypothesis here. But if you work all that lot out and you multiply it together, what you find is that there is the possibility of around about 2,000 sources of life. And then if you multiply that perhaps by 32 stars in the universe, you see that the possibility is that we are looking at an enormous potential source of life in the universe. And Frank Drake has come in for a lot of criticism over the decades. And again, if you're familiar with this, you'll have read it. But actually, at least he had a go at trying to put into simple terms what the possibilities of life may be in the universe. So over the years, this N has been revised downwards. But whatever it is, if we don't look, we won't find it. And of course, this is part of the rationale for the search for extraterrestrial intelligence. Now, some of the background studies that have been conducted, I'll describe now, and these are again by the Seager Lab, who have now started to think on a more scientific basis. What can we do to search for signs of life? And of course, this is the fascinating area of looking at biosignatures and the possibility of being able to find biosignatures on exoplanets. So again, I think both of these papers are also in the public domain. So if you're interested, they are very, very comprehensive and very, very thought-provoking. And of course, the simple hypothesis is actually we can use the starlight itself to characterize what molecules there are, because they leave their own absorption spectrum in the starlight, in electromagnetic radiation from any star. And we can measure that spectrum, and then from that spectrum, we can tell what the molecule is, because of course we've got our own standards on Earth. So it's a very simple and elegant way to be able to measure what biomarkers there may be that predict the existence of certain molecules on planets far, far away. And they started this work in 2016, and it's still going on. And they started thinking about the search for biosignature gases. And they generated a list of all small molecules to start off with using chemical approaches. So they had three filters. Their first filter was, let's think of all the small molecules that there are that are stable and volatile in temperatures and pressure conditions relevant for exoplanet planetary atmospheres. So any molecule that fell outside the range of those temperatures and pressure extremes, they discounted. And of course, this is the benefit of having supercomputers to work with in 2016 and today, which of course you couldn't have done 20, 30 years ago. The next filter that they imposed is then thinking about the detectability, sources and sinks that may ultimately control the molecule's accumulation, and discounting some facets. And then their third filter was thinking about molecules that might otherwise be generated as false positives through some geophysical conditions. And it's a very, very simple conceptual process that should, in theory, lead to a finite and comprehensive list of molecules that could be considered for search for exoplanet biosignature gases. And the reason I really love this concept is that it was put out as a crowdsourcing idea. So they generated these molecules and they said to the scientific environment, these are the molecules that we predict could be indicative of life. Help yourselves, guys, go out and prove it, disprove it, and let's publish it together. And what they did, and again, for the chemists amongst you, you'll be familiar perhaps with some of the sources up here, certainly the Sigma catalog, and this is where they comprise their molecules from. So I won't go through this in great detail, but really what they did is they took a wide range of sources to make sure that the repertoire of molecules that they were including was inclusive and representative as possible. And what they also did is tried not to make it purely anthropic. And by that I mean, they tried to make this as open as possible by considering molecules that are made by life on the right-hand side, but also considering molecules that could be formed and that are formed, but that are not made by life. because of course what they wanted to do is open up the possibility that there are conditions on other planets, other worlds, that we wouldn't find suitable to support life, but they may be able to find suitable to support life. And in a way, this is the concept that they came up with. So our first filter, second filter, third filter, throw all these in at the top, and let's see what comes out at the bottom. And really the output has been quite exhaustive. They found round about 2,500 molecules that should be representative of biosignatures known to be produced by life on Earth. They have put forward those molecules out now into the public domain, and they've asked the question for everybody as scientists, can you now use our database and see if you can detect these molecules in the atmospheres of other exoplanets? And if you can, publish it, report it, let us know, and let's see if we can develop this field together. So I really like this approach. It's yet to yield anything that's indicative of life on other exoplanets, of course, as we know, otherwise we'd have heard about it by now. But I really like the approach because I think it's using a very systematic chemical approach with some assumptions that are called out up front, but it's opening it up to the scientific community. Come on down and make this work or refute it, but let's move the field together. So Sarah Seager in 2013 put together the Seager equation. So this was her version of the Drake equation. And it's a really clever idea because put together in 2013, what she wanted to do is think about, well, what's the probability of identifying an inhabited exoplanet and finding life within the next decade? So of course, that's now by 2023. And the reason that she did that, I think, is to keep the public imagination and public interest going. Because it's great to say, let's think about what we'll find over the next 50 to 100 years, but none of us will be here. Let's think about 10 years. And of course, that piques a lot of interest because hopefully most of us will be here in 10 years' time. And what she started working through is the number of M-class stars with a magnitude of less than 13, i.e. bright enough, fraction of quiet stars, i.e. not too noisy, fraction in the habitable zone, fraction of transiting exoplanets detectable by a James Webb Space Telescope, fraction with life, and of course that's pure speculation, and a fraction with detectable spectroscopic biosignatures. And what she came up with is, well, there's about half the chance of finding an inhabited exoplanet. If we think about our 2,000 now that we showed on the Drake equation, actually it's about 0.5. And David Latham, I think, came up with this wonderful saying, and that's not going to be easy, but we can dream. And then I thought, well, actually cheer up, because if we look on the bright side, this actually doesn't include us. So what Sarah's tried to do here, it may not necessarily give us what we would like to believe is that life will be found within the next decade. What she's tried to do is put this on a level that we can now comprehend and register that's meaningful to us. And by opening up the type of work that she's done within a crowdsourcing environment, I think is spurring a lot of interest to now investigate and study the atmospheres of exoplanets. So what is life? And I thought, well, it's actually a little bit remiss of us not to think about what is actually life itself, because of course we're only looking at it from an anthropic point of view, i.e. we're only looking at it from a human point of view. And I've really tried hard to find a fetus with an iPhone. So if anybody's got one, please let me have it, because that's a BlackBerry, as you can probably see on the right-hand side. And I love that little quote on the right-hand side, if you've got time to read it. But I thought, well, let's think about the basic definition of life: transport, nutrition, respiration, excretion, synthesis, and regulation. And that, of course, is our view of what we see of life. But it can actually be different. And together with Matt Williams, we published a paper a couple of years ago when we started to think about, well, life doesn't have to be flesh and blood and cells and membranes and what have you. Is it possible, of course, given the huge interest in machine-based learning, and I'm not a huge fan of artificial intelligence as a terminology, although I know it's very widely used, but machine-based learning for me is a little bit is a little bit more in the moment, because this is effectively what artificial intelligence is. And I was trying to think, well, are there analogues between life as we know it today for human beings, for animals, for bacteria, and life that we could see with artificial intelligence? And actually, when you come to look at metabolism, growth, life cycle, reproduction, response to stimuli, adaptation to environment, and evolution, there is the possibility that we could configure life with the same definitions in the concept now of machine-based learning. In one particular case, a self-replicating neural network that has been developed and is being developed as we speak. So the possibility that we might have to change our thinking of what is life, I think, is here and now. And I think we will be lucky enough within the next decade to see the definition of life change, and perhaps start to challenge a little bit ourselves what we mean by life. So this was one of Carl Sagan's definitions of life, that of course has been widely published: life, living matter, and as such, matter that shows certain attributes that include all of those things that you can see on the left-hand side of the figure. And what we were bold enough to suggest is that life could actually be now envisaged as a system which is self-sustained and able to learn and adapt to environmental input. And is that a possibility of life as a description in the future? That again, we might want to search for in perhaps the decades to come. So I want to finish on just a couple of things. And of course, one is very topical, and this is the Mars 2020 mission. And as we know, it's part of NASA's Mars exploration program, which includes Perseverance and Ingenuity, an astonishing achievement that Ingenuity has been able to take off in the Martian atmosphere, which is just stunning. And of course, we know that its target is the Jezero crater, and we know that it's there purely to look for, at least in part, maybe not totally, but its real remit is to investigate an astrobiologically relevant ancient environment. And by looking at its surface geological processes, by being able to cache samples for return back to Earth, it does offer the real possibility that we may be able to detect either present, which I think everybody would recognize is highly unlikely, or extant sources of life on Mars that we believe very strongly to have harbored oceans and lakes and rivers and what have you. And then when you come to look at one of the figures that NASA have published, and then you come to think of the fact that it's not too difficult to imagine this as being a potentially extremely rich source of life. If we think back to our earlier description of extremophiles, what I was sharing you about what is found in certain concentrations of salt, pressure, temperature, etc., you would think if there was life there, we will be able to find it. The only thing that I find a little disappointing is that I believe it's not until the end of the decade that we'll be able to have brought back the samples that have been cached on Mars at the various drop-off sites from Perseverance. It would be so nice if we could bring them back earlier, but I'm sure if there's a way, then NASA will find it. So just by way of an introduction, by way of a conclusion, what I'd like to be able to summarize for you is why life is extremely likely elsewhere. And I think it's simply because the same ingredients are everywhere. Life started rapidly on Earth, and we know that we have extremophiles that we can identify on Earth that satisfy an enormous range of conditions, pretty much every condition that we can find on Earth. And there's one caveat to come in a moment. So lab experiments suggest that life at least could be formed from the formation of biological molecules, and we can find some high molecular weight molecules in cometary remnants, for example, the Allen Hills comet, and there are others. Life can survive in extreme environments, we've said that. Does the Earth need to be special? And of course, if you've read the Brownlee manuscript on the rare Earth hypothesis, it would suggest that the Earth is special. It is a special environment where life has formed. It's been protected by the gas giants for cometary bombardment. It's had the moon. It has certain conditions that may well not be common in other planetary systems. And if we look at the TRAPPIST system, for example, that piqued everybody's interest 3 or 4 years ago, it's almost certain that there isn't a planet there that has similar conditions to that we enjoy on Earth. And then, of course, if there are a lot of stars in the galaxy and a lot of galaxies, it does beg the question, isn't it inevitable that we will find life, even if its simplest form, elsewhere? And of course, the universe is ancient. There's lots of time for primitive life to have started elsewhere. But of course, remember the Fermi paradox. And if you're ever interested in a more in-depth analysis of the Fermi paradox, again, this is something that's really interested me over the years. And there's been some very, very elegant studies conducted. So finding Earth-like planets is probably extremely difficult. But using what we know about extremophiles, and also knowing that we're able to detect molecules indicative in life by spectroscopic methods, may mean that ultimately our search for biosignatures indicative of life elsewhere will tell us that there are candidate exoplanets where we should focus our attention, because this is where we believe life is most likely to exist. So, summary, life formed on Earth as soon as it was possible. By statistical chance, life should exist elsewhere in the universe. A systematic search for biosignatures should indicate that we'll find life, or at least planets capable of supporting life. And for me, future biosignature research on exoplanetary atmospheres is really key, to me at least, to decide when and where we send future space missions. And one particular thing that interests me the most is we don't necessarily have to send a manned space mission or a man and woman space mission. Why don't we use a non-manned space mission to explore other planets? We could accelerate very small spacecraft nanoships that NASA has been developing at about the 10th or even the 5th the speed of light. That, of course, human beings wouldn't be able to tolerate. So I think the idea of finding candidate planets to search for life and looking at non-human space missions, for me, is really very important for the future. And then when you think of the first detection of the first exoplanet in 1995, just think how far we've come in the space of 26 years. I mean, it really is astonishing. Where will we be in the next 26 years? So I just want to leave you with this question, which I've tried to sort of seed in your minds as we've gone through. So why should organisms be found on Earth that can withstand 15,000 gray radiation, 403,000 times Earth's gravity, 800 meters per second impact collisions? And also, what I hadn't gone to in much detail is we know that there are organisms, a wide number of organisms on Earth that can metabolize perchlorate. And perchlorate, of course, is one of the major contaminants of Martian regolith. And of course, it's also produced on Earth. But if you take that together, does this support the panspermia hypothesis, Arrhenius's original premise, that life on Earth may in part have come from cometary bodies or other bodies outside of the solar system, and possibly even outside of our universe? So I'll leave that thought with you, and then I think my last part is just to say thanks for listening and any questions. And I really want to thank my colleagues at Mansfield and Sutton Astronomical Society and many astronomical societies who have helped and challenged me. I want to thank people who have sent me some of their publications. Mark Shellhammer at NASA in particular has been very generous in sharing some of his unpublished work. And I'll stop there. I think bang on time, and I'll do my very best to answer any questions you may have.
Well, thank you very much, Martin. That was absolutely fascinating. And what I particularly liked is a lot of your slides had the web links on them. So there will be the possibility for delving a bit deeper at a later point and getting into the detail. Now, I have a couple of people in the background checking for questions that have been coming in. The number of people that we've got on the meeting this evening is probably a manageable size, that if anybody has questions, and rather than fight typing it in, if you want to unmute yourself, and we can just do the questions direct. That might be easier than typing it in, although we do have a couple that have been typed in already. John, do you want to run through the questions that you've spotted?
Yeah, we've got one from Angela Nicholson, or maybe Keith, not sure. At what point do RNA or DNA molecules become life?
Well, that, you know, this is the question that I think, despite all the elegant theories that we have, there isn't anything that makes you think about what would need to happen to construct a simple cell. And I think when we think about even the most primitive cells, what would it take to make the DNA or the RNA part of that cell, the cell membrane, for cells that have a nucleus, for cells that of course have mitochondria, the powerhouse of the cell that produce ATP. And I think if you think about it in its simplest form, when I think about something like a simple protein like insulin, for example, around about 100 amino acids, we know there are 20 amino acids on Earth, today at least. So the chances of those amino acids coming together in the right order to produce an 100 amino acid protein, it is almost impossible to imagine. So for me, there has to have been a nucleic acid world before a protein world, and I would think that that's pretty much accepted. The premise that the RNA world existed before the DNA world, I think, is now fairly accepted too. So at what point does the RNA become life? And I think we would have to imagine the only way for RNA to become life as well would be accidental absorption of the RNA into a lipid system, a lipid-based system, which gives it a membrane, which protects it from degradation. Now, then at some point, of course, there has to be a simple ribosomal structure to be able to translate that RNA into a protein. And trying to think about how to put that together, I think has baffled people for many, many years, because you would think that the chances of it happening by accident would be so remote that it could never happen, and yet clearly it has happened. So it's a question that I don't think there is a serious answer to. Otherwise, it would have been proven in lab-based experiments now. But to me, the order has to be RNA first, and then DNA, and then the proteins that are transcribed and translated from the DNA and the RNA. But there has to have been some form of invagination in their membrane structure that then ultimately has a ribosomal system capable of translating the proteins, otherwise you wouldn't get anywhere. And then, of course, ultimately a nucleus formed to be able to allow the cell division that we see today. So it's an intriguing question, and I don't have an answer for it, but I think that is the hypothesis, that RNA has become invaginated into membrane structures, and that simple ribosomes are formed. And once that's happened, there's been then the production of proteins in the simplest form that allow that life to survive, that very primitive life to survive, and then under the right conditions evolve into what we know today. But conceptually, it's hard to get your head around, I think. it's not the best of answers, I'm afraid, but I think and as far as I'm aware, that is the current answer.
Yeah.
Okay, we have another one here. First of two, the subject of Venus, actually. Is there an extremophile capable of surviving Venus?
Well, as far as we know, there isn't. And I see the other question here, which is around the phosphine component of Venus. And I know this was one of Carl Sagan's wishes, that he thought that life would be found in the clouds of Venus and be able to tolerate phosphine. And as far as we know, there isn't. But then, of course, we've never looked. And until we look, I'm not sure we could rule anything out. I mean, who would ever have believed that you would find an extremophile capable of tolerating such enormous doses of radiation? You would not have believed it. So I think the answer is we don't know that there's an extremophile, but then I'm not sure that we've looked. I'm not sure, I haven't seen any reports of whether or not it's ever been tested with some of the current extremophiles. I don't think so. So I'm assuming that there isn't an extremophile that can tolerate the phosphine concentration on Venus. But I think we have to look, and we have to send craft there, and we have to sample the atmosphere. So we've got one here. Is the panspermia idea compatible with the idea that life is prevalent because it's an emergent property? Yeah, I think it is. And you know, when I think that the panspermia hypothesis was first probably taken seriously 10, 15 years ago, I think it was still sort of laughed at to some extent, because the idea that life could come from elsewhere was almost seen as a little bit alien to the belief that life evolved purely on Earth. And of course, they're not mutually incompatible concepts. Life has evolved on Earth, as we know, but it doesn't necessarily mean that all life evolved on Earth too. So I think the idea that panspermia has contributed to life on Earth is perfectly viable and perfectly reasonable. Of course, the proof in the pudding would be if we can find some form of primitive life on any comets that we've either visited or studied or intend to, or any, of course, that have come to Earth. But then, of course, we've got the problem of Earth contamination with anybody that hits the Earth. So I think that the question will only be answered if we can find primitive building blocks of life on any of the comets that we seek to land on in the future. But of course, the idea of landing on comets in space, I think, is more geared towards space mining rather than finding life. I don't think we can rule it out, but we can't rule it in either, unfortunately, not yet. But it's more attractive. So I was trying to think of, are there other examples of life on Earth that by all accounts shouldn't have evolved to tolerate conditions that don't exist on Earth? And those are the only ones that I can think of: the radiation, hypergravity, and possibly now the effects of impacts. Who would believe 800 meters per second impact? A living organism can tolerate that. I mean, it's just unbelievable.
Can I sneak a follow in up?
Sorry?
Can I sneak a follow into the phosphine?
Yep.
Because we just tripped the last part of it. So there was a big announcement that they thought they'd made it, but they've had to retract it. I just wanted to know if you want to maybe talk a little further on how hard it will be to do these biosignatures on exoplanets if we're struggling with the positive identification of a near neighbor to get a measurement that we'd be sure of an exoplanet. So
I mean, I think you're perfectly right, and it's going to be very tough. But I think as we get more used to it, and as we get better at sifting out the false positives, I think confidence will increase. And I think when you think about biosignatures that are indicative of life, you could almost compile, if you like, your own hit list of the 10 biosignatures that you would want to see before you started getting excited. So in addition to obviously water vapor, ozone as a proxy for oxygen rather than oxygen itself, carbon dioxide, methane, the usual bunch that I said earlier. I think we could start to see areas where you might get excited, again, based on the absorption spectrum in the starlight. But the stimulus to be able to send a mission, and the confidence that we need to send a mission, for me, it's not manned by any means. Even if we could do it, to me, it would be a fully automated space mission, preferably using some of the microtechnology that there is available now, and being able to send it at vast speeds over vast distances. But the exercise is one of public relations. How do you fund something that's going to take 30 years, 40 years to give you an answer, especially if it's going to cost billions and billions of dollars? So part of this, I think, is almost, if you like, getting the general public behind the concept that space exploration is valuable to start off with, but also that it warrants this type of investment. Because for all we would like to do, no nation is going to pay for these types of studies separately. To me, it would have to be a conglomerate of nations willing to pay for it. So if I was to say to everybody here, I want to crowdfund a space mission, would you give me £10 each? I'll probably get a tenner out of everybody here. Perhaps if I'd come up to see you and we'd got a bottle between us, I could ask you for £100 each, and I might, on a good night, perhaps get £100 out of everybody. But if I was to ask you £1,000 each, you'd send me on my way. And I just wonder whether these types of missions for the future, to keep the public imagination going and to accommodate the fact that, as you just said with phosphine, there will be false positives, means that we're going to have a crowd-funded effort across nations and across governments and space agencies. So it's sort of dampening on everybody's parade a little bit, but I think we have to acknowledge the fact that as science evolves, and it's been 26 years since the discovery of the first exoplanet, maybe not within the next 5 years, but 10 years time, 15 years time, I think the advancements in technology will allow us to make these types of leaps and best guesses as to where we should search for life. And I just hope that I'm around to see it, because I think now is a fascinating area for young people in particular to be scientists. I think the opportunities are just astounding. I wish I was 30 years younger, that's for sure, as do we all, I suspect.
Okay, that great questions. Is there anybody else in the audience that has any questions they want to ask? If so, feel free to mute your mic, unmute your mic, and we'll be able to see if you've done that, and we'll let you know that you can then speak.
I can keep the question going if there's no one.
Yeah, go for it.
Okay, Ian, go. While everybody's thinking about it and maybe trying to figure out how they unmute themselves, if you want to go on with your question.
Yeah, if you're struggling to unmute as well, you can always raise your hand and we'll do it for you. So I put it in the chat, Martin. So on the Fermi paradox, just asking, if there's a bit of a conflation between life and intelligent life, and whether there's the sad prospect that we can evolve, exist, and blow ourselves up in the blink of, in the universal blink of an eye, so that life is abundant, but life at any one given moment, there's actually not that much of it in close enough proximity for anybody else to know that it's there.
You are absolutely spot on, and I've got the box up here. Let me just take it off so I can see. Okay, you're absolutely spot on, and when you think of the rise and fall of certain civilizations on Earth, for example, so the Mayans, the Greeks, the Romans, what have you, that is assuming, of course, that we call ourselves civilized, and some people might argue with that on occasions, but let's assume that the average rise and fall of the civilization is around 10,000 years. So there has to be an absolutely perfectly overlapping period of time where our civilization is advanced enough to coincide with an alien civilization in its life cycle, such that we coincide and that we have the opportunity to detect signals and what have you. So if we were to imagine civilization on Earth as it has done in the past, go down the dip and start to decrease in its technical ability, which may happen at some time in the future, if we're out of sync with the rise in technological ability of an alien civilization, then we may never either detect each other or what have you. So I think the difficulty between finding the right coincidence of advanced terrestrial civilizations that have the technology to even understand that they are talking or meeting to each other or detecting signals is a huge confounder. Because of course we can find perhaps life that isn't intelligent enough to even know that we're looking for it, bacterial life, viral life, etc. I think this is part of what's called the soft and hard Fermi paradox that's been worked through for some time. So it is perceived that on average, the rise and fall of civilization occurs every 10,000 years, as we've seen from our own history on Earth. And again, if you the boundaries on that may be maybe an order of magnitude, but even if you have that order of magnitude between 1,000 and 100,000 years, you still have to have that overlap between civilization on a receiver planet, which is Earth in our case, and a civilization on a transmitting planet, which could be somewhere else. So you're absolutely right.
Okay. So is there anybody else that has a question? If you want to unmute or type your question in, this will be the last chance. While I ask Martin a question I've got, I have to say these tardigrades just fascinate me. I think part of it because they caught my attention on some Star Trek episodes many years ago, and the thought that they can survive floating around in space, and by the sounds of it, they could crash land on the Earth and survive the impact. How do we know if we manage to find a tardigrade on the Earth, whether it has evolved and came to life on Earth, or whether it's one of our recent space tourists
that's a good question.
that has arrived here? Is there a way that we can look at them, perhaps their isotopic composition or something like that, that could tell us, did they originate on Earth or have they came from elsewhere?
No, that's a really good question, and I think I think part of the way of doing it would be to at least understand the phylogeny of the tardigrade to start off with. So the fact that we can map where the organism is within the repertoire of our understanding on Earth. So of course that just doesn't rule out that it's not Earth made or Earth formed. Just because we haven't found it, it could be a new species. I think the other element that would give me faith that we found perhaps a space-borne organism rather than an Earth-formed organism is if we see that its amino acid composition is in some way different, or if we see that there is some adaptation to how it's able to survive and metabolize. So if, say, for example, and the isotopic idea is a really good one, but let's say, for example, that we were able to find proteins that were indicative of the existence of other amino acids. That again would be a real signal to me that we're looking at life here that is able to evolve and use protein structure to make life, but actually it's not used the amino acids that we see on Earth. And I just wonder whether or not it's possible to do well, it would be possible to do those studies, of course. The other concept, and it's very interesting because again, a little bit like the phosphine discussion we had earlier, I think it was subsequently retracted because it concerned the existence of arsenic-based life. which I think, if I remember correctly now, was retracted because it was a contaminant that the life form had been found in. But it serves the point that if life is indicative of living off a different substrate, or with a different amino acid composition, or has a repertoire of amino acids that are different from those found on Earth, or perhaps has a different place in the phylogeny of those organisms found on Earth, I think you could start to build lines of evidence that would strongly support, but probably not prove, strongly support the fact that the organism came from beyond the Earth. So I think there are ways of doing it.
Okay, thanks for that. Okay, last chance. If anybody's got a question, pop your hand up, or
I've got one. Ian's put one in on the
All
chat.
right.
Another one.
okay, go for it, Ian.
So I'll read it out as well, but you can see it in the chat. So you've shown how tough the tardigrades and the bacteria and everything are. How do we prevent our robotics missions accidentally acting as panspermia?
Oh, gosh, that's a good one. Oh, my goodness. That's a really good one, because especially, what would they call? Have you come across von Neumann replication probes? So von Neumann replication probes, this was very, very futuristic when it was first put forward, but it's not so futuristic today. So with von Neumann replication probes, what he hypothesized, if we were able to make a self-replicating robotic system and effectively set it free into space, then it should be able to colonize the universe. I think it was within the space of 100 million years, something like that. So the concept was that these automated probes would be able to replicate themselves, and just by the fact that there were enough of them, would be able to find their place in the universe. So that is, if you like, almost if you like, an artificial form of panspermia. So I think with our idea of a robotic system, we would have to have we would have to have the ability for it not to replicate itself, which we can't have, which we would have anyway, because we can't do it today. But I think we would also have to have at least the way of identifying it as being an Earth-made source of life, for want of a better word. So in the way that Gordon was asking about, how can we almost, if you like, genotype or phenotype a foreign body having landed on Earth, I think we would almost have to label our robotic system as, for want of a better word, made on planet Earth, and for any recipient who sees it to see that this has come from Earth. And that we don't artificially, if you like, introduce the concept of contamination elsewhere. But in a way, perhaps it's not such a bad idea, because promoting panspermia with a robotic organism, perhaps there are some positives around that if it could be done, because maybe by identifying it as Earth-made is actually a flag to whoever receives it that this has come from a civilization that's advanced enough to be able to generate this technology. So there may be some positives there, but I think it's a good question. And for at least for one now, we don't have an answer to it. I know that the answers, certainly for machine-based intelligence around robots, have been to build in kill switches, which is effectively to stop the equivalent of an Arnold Schwarzenegger Skynet scenario, where you have artificial intelligence going rogue, if you like. And the military are certainly looking at building in kill switches or stop mechanisms into some of their technology that's in development for warfare. So there might be the possibility of doing that too. But it's a really good question, and I think all of these open up the big ethical debate on fully automated space colonization. Is how do we explore without contaminating? And that'll be a debate that I'm sure will go on long after we've gone. That's a good question.
Okay, excellent. I haven't spotted any other questions coming in. John, have you seen any?
No, nothing from here and nothing from Facebook.
Okay, so I think that's probably a good time for us to thank Martin once again for taking the time to give a truly fascinating talk for us. It is greatly appreciated, and what we're going to do is organise a little parcel with some CATESS goodies to be sent
Thank
out
you
to
very
you.
much. Thank you.
So I'm hoping that you'll enjoy that. I see Christian's popped up on screen. I don't know if you want to add anything, Christian. Christian, well, obviously is the man who's been liaising with you to make all the arrangements for you to give a talk. So thanks to Christian for doing that, and thank you, Martin, for taking the time to do so.
Oh, wonderful. Thank you very much. And if you're ever interested in knowing something about development and discovery of medicines in space, you know where I am. I'd love to talk to you again, but I'm sure you've got many people on your invite list.
We have a list of people, and your name is definitely back on the list for if we can entice you to give another talk. So thank you again for doing that for us.